name is Adeline, and throughout this semester I actually learned a lot of things through our courses Microbiology 1, BMY 2, 101. So I still remember about our first class. During our first class, our author actually asked us about what we know about microbiology and what we want to know about microbiology. So at first I actually thought about microbiology is actually the study about small microorganisms that can actually be seen only by using the microscope, and actually want to know more about the extremophile, about the microorganism that actually can survive under harsh conditions. But throughout this semester actually I have gained the knowledge about that microbiology is much more than that. It is so much more than what at first that I thought about it. So our first chapter was about introduction to microbe. So it is important, since we actually have to have the basic knowledge about microbiology to ensure that we can actually call ourselves microbiologists. And during the second chapter we learn about microscopy. The microscopy is about how there are so many types of microscope, the type how there is different way to staining, like grain staining, differential staining, and so on. So it is important, since us future microbiologists actually have, since using microscope is actually the common thing that the microbiologist will do. So it is important for us to know what microscope there is and how we can actually stain, so that we can obtain the result that we want. And the third chapter was about prokaryote, the archaea and the bacteria, and we also learn about the eukaryote, protozoa, fungi, algae, and we also learn about the viruses. Then Prof teach us about the genetics and epidemiology and public health. And after that we actually learn about the microbial growth and microbial nutrition. It is important for us to know how the microbe can actually grow, so that we can control to obtain a pure culture, and so then also how the microbe feed itself, so that we can actually provide the nutrient and that is needed for the microbe to grow. So we actually learn about the microbial growth, before we actually learn about the microbial metabolism and nutrition, then we learn about the microbial growth, how it is growth in the culture, about the different media. We actually gain some knowledge about it, and then we actually learn about the growth control, sterilization, and disinfectant. This is how we can actually sterilize the microbe, how we can disinfect it, and to prevent any contamination during our experiment. And we also learn about how we can actually inhibit and kill the microbe. And the last chapter was about the antimicrobial chemotherapy. So throughout this semester we actually learn a lot about our knowledge, the basic knowledge for us to prepare ourselves to be a proper microbiologist. So throughout, in conclusion, we actually obtain the basic knowledge, the knowledge for us to know about all kind of microbe, and also the proper technique for us, so that we can actually present ourselves to obtain the result that we need in the lab. So not to forget about all the experience that we gained throughout the trip, to the Nusel, to the Yaku, and to the Nyawa. It actually broaden our mind and broaden our passion for the study of microbiology. So I would like to thank Dr. Wan for giving us so much love and so much attention throughout this semester, and I hope that we will get to learn from you again, Dr. Wan, and thank you for all the knowledge that you have given to us, all the love and all the care that you have given to us. So I would like to thank you, Dr. Wan, and until we meet again. Thank you.